Okay, so remember I've got chat function open at all times. The more you contribute into there, that would be massively appreciated. Just makes the session a little more interactive. So I can find out about what you do know and what you don't know. So first of all, I guess the first question would be, have you used a microscope before? And what we need to do is we need to be able to label the parts of a microscope. So I don't mind in what order, but if you have used a microscope and you know the names of some of the parts of a microscope, if you would pop those into chat for me, please. For a little bit unsure, what I might do is watch a little video clip. Say this is a GCSE required practical. So anything that we learn right now is a bonus. Okay, so we've got a couple, we know a couple of bits. It's good. Like it. If you haven't ever used a microscope, just pop that in chat as well. Just let me know so I know where we're starting at. Good, we've got some idea of these. Right, okay then. So what we'll do is we'll crack on, we'll label this up, and then we'll talk about how we use a microscope. In fact, I'm just going to switch my slides around actually, because I've just had a thought. So we'll do it this way first. Right, let's have a look. Answers that we've already got pinged through then. So we've got, my pen wants to work. Oops. Right, down at the bottom, we've got a mirror. Now, the mirror is used to angle light up into the sample. Okay, so we've got that mirror at the bottom. Now, if it's a nice sunny day, we might be able to use, oh, brilliant. We might be able to use the natural light from outside, or we can use a lamp, like a desk lamp, something like that. And we can use that as a light source and then use that mirror to angle that light up into it. Fabulous. Ah, brilliant. We've got some lovely labels come through here. Well done, guys. So up at the top then, we've got the eyepiece lens. Okay, so try and squeeze it in the box. So we've got eyepiece lens. Where I remember them is that is the lens that I put my eye to. So it's the eyepiece lens. Underneath them, we've got the objective lens. Okay, and this has like a little turret, and it's usually got 2 or 3 different lenses on there. Okay, so it might be something like times 10, a times 5, a times 40, whatever. Super. Here we've just got some clips, and these clips sit on this part, what is the stage, and this, when we've got our slide, when we've got our sample, that's where we sit them underneath the clips on the stage so that it keeps it in place. It doesn't move around. This one, don't worry too much about this one. This one is called a condenser, and I'll explain what it is. So basically underneath, excuse my wobbly circles, we've got a series of holes underneath that get smaller. It looks something like that. So what we can do is we can twist that so we can control how much light is coming up through the sample. So if we've got it on the larger hole, then obviously more light is going to come through. And if we twist it to the smaller hole, we're going to get a smaller amount of light coming through. It just allows us to control how much light comes through. And then we seem to be good with these ones. So we've got here, a rough or a coarse focus. So we've got a focus wheel, and then we've got a fine focus. Brilliant. Oops, let me go back and put a C in focus. And then we've just got the arm. and the arm of it, or the handle, is basically just there to carry it, and under the base, and on the arm, and transporting that from wherever you need it safely. Fabulous. Right, so let's have a look at how we prepare a slide. So this is how we make a slide. Okay, so what I'm going to do is I'm going to flick to the little video and we'll watch how he creates a slide. You could be typing some bits and bobs in if you've made one and you've already got some ideas. So we're going to take a small piece of onion first, but probably you'll pick up from here. This will be the first bit that you understand what he's doing. And then kind of 1, 2, 3, 4. What are the 4 steps that he does to produce that slide? So let's have a look at the little video, because it's better than me talking all the time, isn't it? Right, let's drag this across. Oops, if he wants to move. Sorry, my computer seems to be wanting me on a go slow today. Okay, so you should be able to see and hear this absolutely fine. I've shown plenty of videos this way before, but if you can't, please pop it in chat and I'll make any alterations. So I'm just going to skip it on because first of all, he's talking about the microscope. I think we're about here. It's where he's making a slide.
Objective lenses, we have 3 here. We've got a ×4 lens, a ×10 lens, and a ×40 lens. The light coming up from the mirror up to our specimen here goes up into this lens, up through here to the eyepiece lens. This is another 10 times magnification lens, and then we can see our sample when it's on the microscope. What this means is ×4 ×10 gives us 40 times magnification. That's our lowest magnification, all the way up to ×40 ×10 gives a magnification of 400 times. So this is our microscope. Now, to set up a specimen using our microscope, we need to collect a glass slide. Please carry them like this, to touch the edges only, so avoid putting greasy fingerprints all over our glass surface that would make it hard to see our specimen. We need to collect a bit of onion skin. So get some onion, break some bits off it, and generally if you split a small piece of onion, you're looking to get a thin layer of skin coming loose, and then you try and peel that layer off. You can use your fingers or a forceps. Fingers work just fine though. Here we go, I pulled a very thin layer. That is a single layer of cells. So we've a very thin layer of tissue there. Lay that on your slide. Try and spread it out to avoid any folding or creasing of the sample. Again, it'll make it easy to see. Under the microscope, if there's any creases or folds of the specimen, you'll have 2 layers of cells underneath the microscope that you can see, which confuses what you're seeing. So try and get a nice even layer out. I need to add a couple of drops of iodine. So just 3 drops here should be sufficient. The iodine is a food test, is in another one of the required practicals. The iodine is going to stain starch that's in the cells. Will help us visualize our onion cells under the microscope. I need to add a glass cover slip again. Notice I'm holding it by the edges to avoid putting greasy fingerprints on the microscope slide itself, which again would obscure my vision of my specimen. So lower the cover slip on. Try it slowly, avoiding any air bubbles if at all possible. If at this point you've got excess iodine, excess stain on the slide, just dab with a bit of green paper towel to draw up the excess stain. You can just tap down gently on here if you feel the need to, but it shouldn't be necessary. But if your cover slip is not sitting flat, you can just do that. So you should then have a very thin specimen preparation, glass slide, onion skin, iodine, and then your cover slip. This goes onto the stage. Just place it under the...
Right, I'm going to pause him there because he's explained most of it. So if we just number these steps, apologies. If we number these steps 1, 2, 3, and 4, you just want to tell me anything that you picked up on how he's preparing that slide, anything we must do, must not do, anything that you can remember at all. Just type of it, pop it through in chat, and then I'll start and collate it on here. Brilliant. Right, I'm just going to transfer your answers onto this slide, and then we'll talk about it. Super. Brilliant. What else we got? Super. Okay, awesome. Right, so in step 1 is removing the skin. Okay, now the skin, it's really handy to use an onion because that skin on the inside is just 1 cell thick. And when we observe cells, when we want to observe a sample, we need a really thin sample so that the light can get through it for us to be able to see it. So onion is a brilliant one to use because it's just handy that it's got that thin layer of skin. Super, we're going to then lay it flat on a slide. Yeah, and try to avoid air bubbles. We want to try and get it as flat as possible, because otherwise what happens is that it looks like we can see something where actually we can just see air bubbles. Super. I've also put up there as well precaution. She picked up on that. Try not to get fingerprints on the slide, so kind of carrying it sideways so that we don't get our fingerprints all over, because then again we could be seeing the fingerprints rather than the cells we should do. Super. Place iodine. I'm going to put in brackets there a dye. Iodine is used when we make onion cells, but if you, for example, you can take some skin cells from the inside of your cheek and we use a different stain, a different dye. So we just need to dye them so we can see them. But you're absolutely right, it's iodine for onion. So place the iodine onto the sample, and then we place place a cover slip over the sample. And he said, didn't he, at the end, that if there was any excess iodine, we could use a piece of paper towel just to get rid of that excess iodine. Fabulous. Awesome. Right, okay, so what we're going to have a look at now then is how we actually use the microscope. So what I'm going to do is I'm going to pinch this one that we've labelled up. Let's take the microscope as well. We don't just want the labels, do we? And I'm going to delete that one because we've already named them. There's no point doing it twice. Brilliant. Right, so we'll do the same thing again. We'll watch, and I want you to try and pick out, how do we use the microscope? So we've made our slide. We're going to put the slide onto the stage, but there's going to be a certain way that then we do things. So I want you to try and pick up whereabouts the stage needs to be, and what we do with our lenses. Okay, just a couple of things there to try and pick up from. So where does the where does the stage need to be, and what do we do with our lenses? I'm going to see if I could put this on full screen. It might be too blurry. We'll try. Oh, wasn't working the other day, but it seems all right. Cool. Right, let's go for it.
These clips are used for holding the specimen in place. Might sound silly. Cover slip goes up, otherwise it will not focus. The microscope will not focus properly. So the cover slip is on the upper surface of my glass slide now. So we now have light coming up through the specimen that is now on the microscope stage. It's now to the point that people find most difficult. And this is where people find microscopes very frustrating, as they cannot focus properly on their specimen. So these are the tips you should always run through if you cannot focus on a specimen. So this is what you'll always do when you first use a microscope. But if at any point using a microscope you are finding it hard to see your specimen, run through these stages to troubleshoot that problem. Step 1, make sure you're on the lowest magnification lens, our ×4 objective lens. Step 2, get the stage and that lens as close together as possible. It is good practice to get low down whenever you are moving the stage and a lens. So I'm going to get down nice and low, and using these large dials here, they are called the coarse focus dials. I'm going to move, in this case, the lens is coming down. In some microscopes, you might find the stage moves. It doesn't matter which way it's going. So these are now as close together as possible. Then to look down the microscope and then focus, turn the knobs towards you, going to raise the lens away very slowly. So I'm going to carefully use the coarse focus here. You might find it easy to use these smaller dials. These are the fine focus dials. They move this much more slowly for easy fine adjustments. So I'm going to look down here. I'm going to raise this up until I can see my cells. And that is now in focus. I can see almost like a tiny patchwork of little bricks, little cells, sort of oblong in shape. You can see an outline around them down here. Now they're very small still, it's low magnification, but they are now in focus. It is easier to do it with this small lens. Now I've got them in view, I can increase my magnification. To do that, all you need to do is spin the next objective in, get down low, make sure it's not going to touch your slide. So again, there's lots of space there, so they're not touching, it's not going to damage the lens or the specimen. And it should roughly still be in focus. Now I need to just adjust that quickly. So just very small adjustment. In this case, I'm just bringing the lens up very slightly. Has brought it back to focus, but I have not changed it very much. I think that's a problem people have, is they make big changes to their focus. They change these dials quite a lot. They move the lens a lot whenever they change the lens. It was a very, very small adjustment. So I now have the specimen.
All right, I'm going to leave him there. All right, so any of those, do you call them steps? You say step 1, do this, step 2, do that. Any of those points, what key points was he making? What did you pick up? And just pop them in chat for me, please. Good, yeah. This is awesome, guys. Well done. Brilliant, yeah. So put a slide under the clips. Start with the smallest objective lens and bring the lens and the stage as close as we can, but just looking at it, making sure they don't touch. Brilliant. I'm going to run out of room. Super. So we're going to turn then the focus wheel, and we're always going to turn it so that the gap between the lens and the stage gets bigger. Okay, we're never going to wind it so that they're coming back close together. We do that while we're looking at it, getting down low and looking at it, and then we wind it to get it in focus by moving them further apart. Once we've got them into focus, we can then increase the objective lens. So we can increase the magnification by changing the objective lens. Amazing, guys. Well done. Right, now, thinking about magnification. So he did talk about this in the clip, okay? Now, when we're thinking about what magnification is that microscope producing, all we have to do is we have to think about what is the magnification of the eyepiece lens, and we multiply it by the objective lens. So if we have a quick look at this question, these are dead straightforward on it already. So we've got eyepiece lens of times 10 and an objective lens, so we've got the numbers from, of times 40. So we multiply those two together and we get a total magnification, whatever we're looking at will be 400 times bigger. That's the easy part of the magnification calculations. We're gonna make them a little bit harder in a minute. Right, so I'm just gonna put you 3 quick questions on the screen. So we just need to look at what eyepiece lens is he using. What is the objective lens, so therefore what is the total magnification? You can just put number 1, the answer, number 2. Oh, smashing these. Cool. All good answers so far. Amazing, amazing. So here we've got the same again. We've got 10 times 40. So we've got a magnification of 400. Here we've got 20 and 100, magnification of 2000. And in our final one, we've got 10 and a 30, got a magnification of 300. Awesome, well done, guys. Right, let's take it up a little level then, and let's have a look at how we work out the magnification of an image that we've got, because I'll show you how these relate to some GCSE type questions. Okay, so this is the kind of thing, this is all, if you've used a microscope, this will all build up, and when you get into GCSE, it becomes one of the quite required practicals. Okay, so here's our equation up at the top. The magnification is the size of the image. So the image is what you're looking at. So that might be what you're looking at down the microscope, or what photograph you've taken, or what's in the textbook, something like that. So we've got magnification is the size of the image divided by the real size. Now, I don't have a virtual ruler, something I need to look into. So what I've done is I've already pre-prepared and pre-measured them for you. Okay, so with this then, the way that I do any calculation, and we'll get into good practice of doing this, any calculations are sort of, I'm going to put them into symbols. So I'm going to have magnification is image over actual. And then what I'm going to do is I'm going to down the side write down the bits of information that I know. We're not missing an action, don't worry. So magnification, it asks me this, calculate the magnification. So I'm going to leave that as an M. So that's the one I don't know yet. Do I know the image size? Okay, now I've got to be careful and read which one it is. This is the real size, the size of the cell dividing in the book. So the image is the one from the book. So that is 100 millimeters. The actual, the real size is 0.05. And then I'll just substitute it into my equation. Yeah, guys, you're smashing this. So you're going to have 100 divided by 0.05. Okay, got a calculator handy? Or have we just got super brains? Got one answer through. Any more for any more? Okay, so it's 2000. So that cell here has been magnified 2000 times. It's 2000 times bigger than it is in real life. Okay, fabulous. Great stuff, guys. Well done. Well done. Okay. So let's have a look then. I'm going to jump around a little bit. Makes sense. I'm going to go to this one because this one is quite nice. So this is a GCSE question. Okay, so pushing you a bit because I think you can do it. So I do exactly the same again. Pick out my bits of information. I want to know magnification. So do I know the image size? Do I know the actual? So I'm just going to be quiet a minute and let you just have a look and a think. Have a go if you can. And then I'll go through it with you. Good question. Don't know, I guess if somebody wrote here magnification, you need 2 lines, wouldn't you? So there's only really one extra line. Don't know, don't be fooled by exam questions. The more you look at them, the more you'll realise they give you loads of space to write, and half the time you don't need all that at all. Right, let's have a look then, because I've got a couple of answers pinged through. So image was 40, actual, so the real actual size, 0.1. Magnification, the image is 40 divided by 0.1. You guys are absolutely spot on there. It's been magnified 400 times. Awesome. Right, let me see if I've got another little one like that we can do. Right, I'm going to put this this little question grid on. Okay. Now all we're going to do, don't get too giddy, because some of them ask you to calculate magnification, some of you ask you to calculate other things. So all we're going to do is I'll number them. We're going to go for number 1, 2, and 3 to start with. Okay, so I will be quiet for a couple of minutes, let you have a read and have a go at them. And we'll go through those. And then if we're still absolutely smashing it, we'll take it up a level. We'll do a little bit of rearranging. I am impressed at these guys, well done. Right, so magnification equals image over actual. Okay, we want magnification, so that equals the image. Image size is 2 millimeters divided by actual, which is 0.01. And that is 200 times magnification. Well done, guys. Okay, magnification, image over actual. We're only calculating magnification in a minute. So the image size is 10. And the actual, sorry, I just did a very strange noise and wondered what it was. 0.02. And so we've got 5,000. How's your ace at this? Magnification image over actual. Okay, so this one was a little bit sneaky because it's been giving you the numbers the other way around. Okay, so this one says a red blood cell has a diameter of 0.007. So that's real. And an image is 3.5. A bit sneaky, that one. It told you the numbers the other way around. That's why I say always pick them out, pull them out of the question. 0.07. And that is 5,000 times bigger. Awesome. Okay, guys, I've got a couple of little questions that I want to show you and have a go at. And then if we've time, we'll have a look at how we rearrange this equation a little bit, but we've already pushed into GCSE. Absolutely fabulous. Okay, so let me just pick which question I want to do with you. We're going to start here. Okay, so we may see questions like this. And again, it's just, this is a GCSE question. Okay, so it's giving you the steps of basically using the microscope to look at the specimen that you've, that you've already made. So you just need to put those steps in the correct order. First one's been done for you, so we start with D. We prepare the microscope slide of the cells. And then what's the order of the other 4 steps? Amazing answers so far. Oh, brilliant. So D, prepare the microscope slide. It's done for us. Okay, then we're going to A, place the prepared slide on the microscope stage. Once we've put that on the stage, we're going to use the objective lens with the lowest magnification, so E, fabulous. Then we're going to turn the focus wheel, so B, turn the focus wheel to make the image clear. Once we've got a clear image, we can then change the objective lens to a higher magnification. 3 marks in a GCSE, that, guys. Oh, smashed it. Smashed it. Right, these two then. So the second part, B, is roughly the same idea. You're just going to put the steps into order. And the top one is asking you, what combination of lens do we need to produce a magnification of times 400? Guys, these are GCSE questions. This is awesome. top one to get 400, we need times 10 times 400. You're too good at this, you guys. Okay, amazing. Right, so this time it's slightly different, but same idea. Cells are squashed into a thin layer because this time the previous one top that the slide was already ready for us, didn't it? Cells are squashed into a thin layer. We're then going to add some stain to it so that we can see them. I'm going to put the prepared slide onto the microscope stage. Remember, we always start with the lowest powered objective lens, so that is D. Focus the image, and then we can turn it up to a higher power. Awesome. Right, should we have one last little challenge? I'm feeling confident with you. Right, I'm going to flip to this. And we're going to focus on, I think which one to do. So we're going to look at image. Okay, we're going to look at this one, number 3. Okay, so remember, magnification equals image over actual. So this time we've got to rearrange our equation a little bit. So I'm going to just follow the same process that I've done before. I'm going to write down, what do I know? So it says, what is the image size? So I don't know the image size, so I'm going to put that as I. What's the image size of a virus head? If the actual size is 6.8 millimeters, so I know that, and it's been magnified by 2,500. I'll just leave that with you a second, see if you know what to do in that rearranging. If you do, tell me what calculation you do to work that out. Too good, you are too good. So if I put my numbers into my equation, this is the way that I always do maths. So if you continue to come to my sessions and you continue to sort of work with me, this is how I will always set out the maths, as easy as it may seem, because then if you get into that good practice, when you get to more complicated maths skills, it will help you out. Okay, so I pick out the bits of information I know, substitute them into my equation. So if I follow my equation, it tells me that magnification equals image, which I don't know, divided by actual, which is 6.8. And I want to get I on its own because that's what I want to calculate. So I'm going to take that 6.8 to the other side of the equal sign. So it becomes the inverse. So it's divide on this side. So it becomes times on this one. So what I've got now is 6.8 times 2,500. That gives me a image size 17,000 millimeters. Good, yeah, you all got there, super. Yeah, right, let's just do one because I don't want to keep you too long. Let's do one where we change it around. Let's have a look for a nice straightforward one. So we want to find the image. Let's do this one, number 4. Do that one. Let's leave it on a massive high. So good. Too clever, aren't you? Magnification is image over actual. I'm going to write down here because I'm sick of trying to squeeze it into that box. So what we've got is we've got 200 times magnification. We want to know the image. We know that the actual is 0.05. Let's take it to the other side of the equal sign, so it becomes the inverse. 0.05 times 200 equals I. And pop that in my calculator, and I get 10 millimeters. Oh, boom. Great stuff, guys. Guys, you've been absolutely fabulous tonight. Thank you again for participating. Hope you found that fun and useful. And hopefully I will see you again next week.